Hello, students, and welcome to a screencast on macromolecules. So this is part of Unit 2, dealing with biochemistry. This is on carbohydrates, lipids, proteins, and nucleic acids. We got to do a fun lab on proteins dealing with the enzyme catalase, and we'll test for these macromolecules next class period. All right, so let's dive in. This is going to be a longer screencast. You definitely want to break it up into sections as you answer the questions. Here are my questions for you. Some of them are simple, like factual recall, and other ones are asking you to really dive in. So the first one, what elements are in each of the following? Carbohydrates, lipids, proteins, and nucleic acids. The next one's going to say, a huge theme in biology is structure leads to function. How does the structure of each of the following lead to its function? If this problem is giving you trouble, we're going to do a foldable with it in class. And you might be watching after we've already done it, and hopefully you can answer. So carbohydrates, lipids, proteins, and nucleic acids. Within lipids, you should be able to differentiate between the structure and function of triglycerides and between phospholipids. Then I want you to compare and contrast monomers and polymers. So give an example of a monomer and give an example of a polymer. And then this is really getting at, well, how are they made? Compare and contrast dehydration synthesis and hydrolysis. Then bonus, what makes all the amino acids different? And then it seems like my stuff is hiding everything. Here we go. Describe the levels of protein folding. Hint, there are 4 levels. All right, so see if you can get that. Okay, here we go. So this is just a chart giving an overview of the macromolecules. You do not need to memorize this chart, but just to show you, carbohydrates are going to be your quick energy. They're going to be your sugars. They're made of carbon, hydrogen, and oxygen. You get them in grains, fruits, vegetables, bread. Lipids are going to be, I mainly think of them as fats and steroids and phospholipids. They don't dissolve in water. They're nonpolar. They help to make up the cell membranes in our body, and they help to store a lot of energy. Proteins do almost everything. So proteins, when you're looking at me, you're looking at proteins right now. They help to move our muscles. They're going to help to make structural components of our body. They're going to help to make carry things around in our body. They're just going to do tons of stuff. They're going to serve as enzymes. And in nucleic acids, well, we all know the word DNA. We see it used everywhere, and it's going to be how we transmit our genes to our children. And so let's get into it. Okay, carbohydrates. So we went over a 1 to 2 to 1 ratio. For every 1 carbon, there's going to be 2 hydrogens and 1 oxygen. This holds for the monomers. Once you get up to the polymer level, it's going to be a little different because they did dehydration synthesis. But you should know for your monomers, for like glucose, 1 to 2 to 1. In water, they're going to take this ring-shaped structure here. They provide quick energy. They're made through photosynthesis, and they usually end in OSE. So that's just kind of a trick. If you see OSE, you know it's a carbohydrate. All right, here's my more in-depth slide. Mono means 1, and saccharide means sugar. So this means 1 or simple sugar. Examples would be glucose and fructose. Disaccharides are when you join 2 of them together, and we do that by dehydration synthesis. We'll go over that in the next slide. So examples would be maltose and sucrose. You don't need to know this word. Poly means many, so many saccharides or many sugars put together can be a polysaccharide. I skipped oligosaccharides if you watched the video though. These can be storing things like starch in plants like potato and glycogen in animals. Or it can be structural. It can help to make cell walls. It can help make the outsides of certain arthropods and insects. So let's go over polymerization. So here are my monomers. These are my Lego building blocks. My kids love Legos. They put them together, and then you get your polymer. So every time they do it, I tell them, look, you've made some polymers. And then they look at me funny. Here's how it actually works. So it's going to be called dehydration synthesis. And this holds true for carbohydrates, lipids, proteins, and nucleic acids. So pretty cool. Think about the word dehydration. It means to spit out a water molecule, and synthesis means to make. So we're going to put 2 together, and we're going to spit out a water molecule. Let's see it happen. Here we go. There's our H2 H's, so H2O, and then it's going to be bonded on that O. And here you can see the resultant molecule. So we've combined together these 2, and we spit out an H2O. Now, hydrolysis is just going to be the opposite. We're going to break them apart. To break them apart, sorry, this was an animated GIF. To break them apart, we're going to use some energy and we're going to put a water molecule in, and it's going to break them apart. So hydrolysis, hydro meaning water, and lysis means to break. Okay, so that was carbohydrates. Here come lipids. So lipids are made of the same elements, carbon, hydrogen, and oxygen. So this is a great example of how the structure of a molecule leads to its function. Even though it's made of the same elements, they're going to be very different than carbohydrates. That's because they have these long hydrocarbon tails. These fatty acid tails are nonpolar. They're full of carbons and hydrogens. Every time there's a bend, there's a carbon. And at each carbon, they're attached by a bunch of hydrogens. And they're going to be nonpolar. They don't dissolve in water. They're going to store twice as much energy as carbohydrates. This is good for us, right? Plants can store things as starches, etc. But we need to be able to move around and run around. By being able to store twice as much energy in a kilogram of fat, which would take 2 kilograms of carbohydrates, it enables us to move around quicker. The problem for modern day humans is that we tend to store too much, right? Obesity is on the rise. So here I went over structure leading to function. Carbohydrates with those H's and O's on the outside of the ring are easier to break down than lipids, which have those hydrocarbon tails with lots of carbons and hydrogens. So carbohydrates will be used for energy first. Let's get a little bit deeper into lipids. Here are triglycerides. You've probably heard that word before. So here's my glycerol molecule. It's going to be the backbone. And here are the the 3 fatty acids. So we combine those together. Look, it's our friend the dehydration synthesis reaction again. We combine them together. It's called an ester linkage, but you don't need to know that. You just need to know dehydration synthesis puts them together. And now we have this great molecule for storing energy, and it's going to be a fat. If it's all single bonded, it is a saturated fat. If it has double bonds, it's going to put kinks in it, and that's going to be unsaturated. Typically, unsaturated fats are healthier for us, unless they've been hydrogenated, which makes them a trans fat. Here's a good picture of a saturated fat. You can see all the carbons are surrounded. The carbons are black by hydrogens. Here you have a double bonded carbon represented by this second line here, and it's going to make it kink or make it just kind of wavy. Your cell membranes have both of these in them. The kink helps to keep it fluid, and the saturated fat helps to give it some structure. All right, here comes an animation showing dehydration synthesis. It's going to use an enzyme. Here comes the water molecule out, and these guys are going to get combined. That's how the fatty acids are made to put onto the glycerol molecule. So let's say we're exercising a lot. We don't have any carbohydrates in us. We need to break down this fat. We're going to use hydrolysis. We're going to break off this triglyceride. Then we're going to further break that down and be able to be used by our mitochondria in a process called cellular respiration to make energy. Here are phospholipids. So we're going to have 2 fatty acid tails. Here's still our glycerol molecule, but then we add a phosphate. So instead of the 3rd fatty acid tail, when we have 2 here, we added a phosphate. That's going to give it unique characteristics. This part's going to like water. It's called hydrophilic, the element for loving water. And this is hydrophobic. You've probably heard phobia before. It hates water. So this is going to form with the hydrophilic heads pointing towards the outside of the cell, towards the water, right? Where our majority of our body's water. The inside of the cell has a lot of water in it too. And so these hydrophilic ones are going to point towards the inside. It's a double layer membrane. This doesn't take any energy to make. This is how they naturally want to form. So it's great for our membranes. Okay, steroids, I'm not going to put on a test. They're characterized as lipids because they're also nonpolar, but they are slightly different. They have different functions. You can kind of look at it, though, and go over, hey, are the structure leading to the function. The other lipids that we talked about, triglycerides and phospholipids, don't have these 4 rings. So therefore, they're not going to act like estrogen or testosterone or cholesterol. So can you sit back and think, how do lipids show structure leads to function? I want you to really think about triglycerides versus phospholipids. Think about how do they show different structures, and how does that lead to different functions? You should be able to say something about that phosphate giving phospholipids a different one. So go ahead and review back in the video and see if you can get it. All right, take a deep breath. We got through carbohydrates and lipids. Now we move on to proteins. Proteins are ubiquitous. That means they're everywhere. Proteins are really cool. They can be enzymes. They can help build muscles and bones. They can help fight infections. Right? They do all kinds of stuff in our body. Notice how they're also made of carbon, hydrogen, and oxygen with the addition of a nitrogen atom. So that's going to make them slightly different. So different structure, different functions than the carbohydrates or the lipids. Here we go. Here's what an amino acid looks like. It's going to have an amino group. It's going to have, this is called a carboxyl group. You don't need to know that. It's going to have a central carbon, and then it's going to have an R group. The R groups are different for each amino acid. There's going to be 20 different amino acids. We get them from what we eat, and some of them are synthesized by our body. Every single one of these amino acids has this part identical, but the R chains are different. This is called the primary structure of a protein. So this is the order of the amino acids. We'll do a lot of coding of this later on. Here's another example of it, amino acid with the R group. Here are some different R groups. Lysine's amino acid, glutamine, aspartate, alanine, cysteine. This one has sulfur in it, so it's going to want to bond with other cysteines. It's going to make bridges in our proteins. Here's just what the 20 different amino acids look like. So remember, they would all have the same top part, and then these are the R groups that come off of it. And so these R groups are different. Okay, if you're still with me, here's the R group. It's just in this picture, it's facing towards the top, same thing. Notice how when we join them together, we join together these two amino acids to make a polypeptide or to make a protein, we're going to spit out a water molecule. It's once again a dehydration synthesis reaction. Dehydration, the water coming out, synthesis, the two amino acids coming together. Here we go. Here's an animated GIF of it. This enzyme is going to help remove the water, and then it's going to form a bond. It's called a peptide bond right between the carbon and the nitrogen. If you're counting your valence electrons, nitrogen wanted 3, and so it's going to be happy. It's going to bond with that carbon. Oh, here it comes again. So nitrogen is bonded to this carbon, this hydrogen, and this carbon. The carbon over here has 4 bonds, so it's happy as well. All right, hydrolysis, same thing. So we're going to break down that peptide by inserting water and using some energy. So why would we want to break down proteins, right? You ate that veggie burger or that steak for lunch. We're going to break it down so that then you can use these amino acids to make your own proteins. So we have to break them down before we can use them in our body. The secondary structure. So as these proteins are being made, they can form natural alpha helices and beta pleated sheets. These are due to hydrogen bonding between the backbones of the amino acids. I'm not going to test you too much on this. I'm going to change this from a need to know to a nice to have. We modeled this with the protein cutouts in class, the little tubes that you all flexed all around. What I more kind of want you to get is here, the R groups, those things that I said were hanging off at the bottom, they can be acidic, basic, polar, nonpolar, or contain sulfur. That's going to make them want to form different types of bonds. They're going to want to interact with each other. Here's a way to think of it. All of you all lined up in class would be the primary order. All right, be the order of students. Then the secondary structure, I could put you into different shapes, either helices, or I can make you do the wave. And then I'm going to say, hey, start hanging out with your friends. Like some of you all have bonded together from the swim team and you're more likely to hang out. Others of you all might be bonded more together from the drama department. Others of you all might be more about volleyball, and you kind of gravitate towards each other. And those are going to be your R groups. Maybe you might be kindred choir, etc. Your different tendencies, the primary order being you, the students, secondary order being some shapes I put you in, but then I give you free reign to start folding together as a class. Your different interests are going to make you come together in different groups. It's the same thing for the R groups. And lastly, if we combine multiple polypeptides together, it's called quaternary structure. So if we take my class and we take Mr. Spillis's class and then have you all hang out, you would get a quaternary structure. So that's a lot of information right there, and it's a little intense. You read it in your book, you watched a video, you worked with me in class, and we're going to do a couple more models with it, but this is something really kind of to go through and just keep on practicing. So the way they fold and the way they make these shapes give proteins their functions. All right, last but not least, DNA. So DNA is going to have a sugar phosphate backbone, and then it's going to have a nitrogenous base on the inside. It's quite a story of figuring this out. We'll go over that with Rosalind Franklin later on. These sugar phosphates give you a strong backbone, and then the bases are interestingly held together by hydrogen bonds, which is great because it means that enzymes can open them up to read it and to sequence it and to make new copies, but it's also strong enough to stay closed on its own. So the structure of DNA are these backbones and these base pairs on the inside give it its function of storing hereditary information. It makes it a safe molecule that can coil up and save space in the shape of chromosomes, and it can also copy itself as you make new cells. Every cell in your body has the exact same DNA, except for some of your reproductive cells, but they're going to transcribe different genes. So not all of our genes are on at the same time. I'm not going to worry too much on this test about ATP or RNA. ATP is the energy currency in our body. Here it is down here. It's classified as a nucleotide because it has a base right here, a nitrogenous base. You can see the ends, the nitrogen. It has a sugar, and then it has phosphates. So that's really what a nucleotide is. A nucleotide is a sugar, a phosphate, and a base. Well, ATP has it. It just has more phosphates. And when you get rid of one of these phosphates, that's going to provide energy for something to happen. And so ATP is energy. When it's used up, it becomes ADP. We'll do a lot of work on how does this happen. Here you can see the difference between RNA and DNA, just in case you were curious. Deoxyribose nucleic acid, so you want to sound really smart. It is missing an oxygen. D means without, so without the oxygen, here it has an oxygen in this sugar. All right, how's it done? Well, they're also linked together by dehydration synthesis. Here's my example. This nucleotide linking to this nucleotide, it's going to combine between the phosphate and this part of the sugar, and notice how it kicks off a water molecule. It kicks off this OH and this H, and it kicks off a water molecule. So once again, carbohydrates, lipids, proteins, nucleic acids, all put together by dehydration synthesis. So this part we didn't really get to a lot, so I'm not going to put directionality or anti-parallel on it. But just like a highway, one part of DNA runs upstream and the other part of DNA runs down. This is going to be important when we're reading DNA and when we're copying it later on. And this is just due to the chemistry of how we can add new parts onto DNA. We'll revisit this post the winter holidays. Okay, students, this was long and definitely complex. So what I would really encourage you to do is to go back and see if you can answer these questions, to check your foldable that you're going to make in class on how the structure of the molecule leads to the function. Think about the models we use with the ball and stick models. Think about the Bozeman science video, and go through the POGL that will go in class as well. All right, good luck as you keep studying.